Hello everyone, my name is Nitish Maswine. Today I'm speaking about sexual reproduction in flowering plants. First, let's talk about the flower. It is the modified shoot, also develops from floral primordia. Primordia develops first into bud, floral bud, and then it turns into flower. It is the reproductive part of plant. Androecium is the male reproductive uh, whole consists of stamen. Gynoecium is female reproductive part of flower consists of carpels. Parts of flower. Uh, Here we can see the stigma, anther, style, ovary, receptacle, ovule. Um, Anther and the filament is known as the stamen, which is the androecium, um, and then stigma, style, uh, ov- ovary and ovule makes the gynoecium. Stamen, male reproductive unit, consists of parts, filament and anther. Filament is attached to thalamus or petal. Then here is the TSS of anther. You can see here are the pollen sac, and here from here the filament originates. Structure of anther or microsporangium. Um, it is bilobed and dithecous. Uh, a longitudinal groove separates the theca. Uh, uh, in a cross-section, anther is a trigonal structure consists of four microsporangia, uh, two in each lobe. Later, two microsporangia of each lobe fuses as a pollen sac. Structure of anther. In microsporangium is circular and surrounded by four layers. These are epidermis, endothelium, middle layer, tapetum. Outermost layer protects the pollen and helps in dehennancing and of anther to release pollen. Here is the structure of pollen. Microsporogenesis. The process of formation of microspores from an pollen mother cell to meiosis is called microgenesis. The cells of sporogenous tissue undergo meiosis to form microspore tetrad arranged in cluster of four cells. Each cell of sporogenesis has potential to form tetrad, so each cell is a microsporous mother cell, (MC). On maturation and dehydration of anther, the spores dislocate and develop to pollen grain. Pollen grain release with the dehenance of anther. Pollen grain, male gametophyte, spherical in shape, have two-layered wall, out, uh, outer hard exine layer, in inner thin intine exine, made up of sporopolylene, resistant to organic matter, withstands a high temperature, acid, alkalines, and enzymes. It has a chromatin aperture called germ spore, where sporopolylene is absent. Time. It is thin continuous layer made up of cellulose and pectin. Pollen grain and cytoplasm surrounded by plasma membrane. Generative cell. Mature pollen grains has two cells, one vegetative cell, second generative cell. Vegetative cell. Bigger, abundant food reserves, large irregular, irregular nucleus, um, and generative cell. A small, spindle-shaped and dense cytoplasm and a nucleus floats in vegetative cell cytoplasm. In 60% species, pollen grain are shed in two-cell stage, whereas 40% species shed in three-cell stage, in which generative cell divides mitochondrially into two male gametes. Here is the picture of embryo sac. Female reproductive unit, pistil. Pistil is the female reproductive part of flower. It may be mono, bi, or tri, or polycapillary, syncarpous or apocarpous. Each pistil consists of ovary-style stigma. The ovary has one or more cavities called leucos. Placenta in leucus bear ovules. Number of ovules may be one or more. Ovule arises as primordium on placenta. The short stalk which attach ovule with placenta is conchoid. The primordium grows to a mass of cell forming nucleus. The body of ovule. Two protective covering of nucleus is pugament except at the tip giving a small opening called micropyle. The basal part of ovule is called chrysalis that lies opposite to micropyle. Cells of nucleus uh, uh, are rich in a reserve food, a single embryo sac, or uh, a female gametophyte located in nucleus, which is developed from megaspore. Megasporogenesis and development of female gametophyte. One of the nuclear cell in microphylla region differentiated to megaspore mother cell. The cell is larger, contains densely cytoplasm and prominent nucleus. It undergoes meiosis, forming four haploid cells called tetrad. Three megaspores degenerates and only one megaspore functional. Here are the megaspores which get then degenerates and then develops into embryo sac, undergoes mitosis, then second mitosis, or the third mitosis, and forms the embryo sac. Megasporogenesis and development in female gametophyte. Functional megaspores, the first cell of female gametophyte. Its nucleus undergoes mitosis and two nucleus move to opposite pole and form two nucleated embryo sac. To successive meiotic division, each one of these two nucleus form an 8 nucleated embryo sac. Devices for autogamy and simultaneous maturation of an anther and pistil. Anther and stigma lie closely to clistogamous flower, oxalis communella, ovula devices for synogamy. Unisexual flowers and dioecious plant pollen releases and stigma recipient are not. Uh, Synchronized different orientation of anther and stigma of flower self compatibility. Adaptation of wind pollinated plant flowers. Pollen grains are light, non-sticky, dry, sometimes well exposed anther, large feathery stigma, flowers arranged as in ovule. Adaptation of water pollinated flowers seen in submerged flowers like Vallisneria and Hydrelia and Zoroasteria. In Vallisneria, male flower releases on water surface and female flower releases the surface for pollination. In grasses, pollen grains are long ribbon-like and carried passively to submerged female flowers. Muciliae, coated pollen grain. Adaptation in insect-pollinated flowers. Large, brightly colored, and so uh, snowy. If flowers are small, grouped into inflorescence, highly fragrant, produce nectar, sticky pollens, and stigmatic surface, uh, provide rewards to animals, pollinators such as nectar, food, pollen, or provide safe place for laying eggs. Pollen pistil interaction. Recognition of compatibility pollen is interaction between chemical compounds of pollen and those stigma. Uh, Germination of pollen and development of male gametophyte. A compatible, compatible pollen starts its germination stimulated by certain secretion of stigma and until grows out through one of the germ spore. Content of pollen moves into the tube, that is vegetative and generative slash to male gametes. Pollen pistil interaction. Pollen tube grows through the tissue of stigma style, uh, uh, creating enzymes to digest them and enter ovule through microfile. It enters the embryo sac through um, filiform apparatus of one cisgendered to liberate male gamete. Uh, Germinated pollen grains with pollen tube carry vegetative nucleus uh, to male gametes in fully developed female gametophyte. Double fertilization. Double fertilization is just of uh, two gametes from pollen tube into cytoplasm synergid. Fusion of one male gamete with egg cells uh, in gamete and forms zygote 2N, and which develops into embryo. Fusion of second male gamete with a polar nucleus of central cell to form key chain structure. As synchrony and a triple fusion occurs in an embryo sac, the phenomenon is known as double fertilization. Central cell with pen is called P, which develops in androecium. Post-fertilization events: development of endosperm, development of embryo, maturation of ovule into seeds, maturation of ovary into fruit. Endosperm. Its uh, development precedes embryo development. There are three methods of embryo development: nuclear cellular heliobiology. Uh, in nuclear type, pen divides mitocally without cytokinesis, and endosperm is prenuclear. Then cells wall form starts and periphery of endosperm becomes. It provides food to the developing embryo. If endosperm is completely utilized by embryo, seed is non-ambiguous and present seed is ambiguous. Develop, embryo development. You can see a picture here. Here is the endosperm, nucleus, nucleus, microphyll, hypophyse, valvular heart, forms a heart, then tropoid, then cytodion, and mastoid. Embryo. It starts after certain embryo formation. Zygotes divide mitocally and forms preembryo. Then it develops into globular and heart-shaped and then horseshoe-shaped mature embryo. One or two cyteledio. Special reproduction. Apomixis or agamiosperm. Seeds are formed without fertilization, may develop into diploid egg cell and develops into embryo without fertilization. If cells of nucleus may develop into embryo and pushes into embryo. Special reproduction and polyembryony. If more than one egg may form in embryo sac, it is more than one embryo sac formed in ovule. Other cells like uh, Kindred or nucleus developed into embryo, that is orange, lemon, mango, onion, groundnut, etc. Thank you for listening my PPT.